if at all this dideoxynucleotide triphosphate is added to the DNA sequence, the chain elongation terminates at that particular point itself. Okay, so this is what is being used in this Sanger sequencing method. So let's see how the procedure goes, right? So first of all, what I will be doing is I will be taking 4 different test tubes, right? So I'll be taking 4 different. Yeah. So I'll be taking 4 different test tubes. So let me denote it in different colors. So in the first test tube has what? The first test tube I will be adding dideoxy ATP. The second test tube I will add dideoxy CTP. Third test tube I will add dideoxy GTP. And fourth test tube I will add dideoxy TTP. Okay, so I will be adding dideoxy nucleotides of every nucleotide in 4 different. So 4 bases and 4 test tubes I will be adding. But otherwise, otherwise I will add all the requirements of the PCR in all the 4 test tubes, like the template strand and primer and the deoxynucleotide triphosphates, all of the 4 deoxynucleotide triphosphate, all of which I will be adding in all 4 test tubes. But dideoxynucleotide alone, 1 test tube, one dideoxynucleotide I will be adding. Understood? So this is how I will prepare my test tubes. Next, I have the template strand now. Yes, you have the template strand now. So what is this template strand? Suppose I have CTAG, CTAG. This is what is the template strand that I have. Now, if this is subjected to PCR, what happens? Complementary strands would get synthesized, is it not? So while synthesizing complementary strand, you will be adding the nucleotide that is complementary to this sequence, right? So in the first test tube, when you're going to run the PCR, what are the fragments that you will be getting? Okay, let's see that. So in the first test tube, when you are running the PCR, I will be getting, so what is opposite to G, C, what you will add? G, right? Opposite to T, you will be adding A, right? So in this point, the procedure adds deoxynucleotide, deoxy ATP or dideoxy ATP. If dideoxy ATP is added at this point, the chain elongation terminates at this point, right? So what fragments you will get? I will get a fragment size of say 2. Okay, so only 2 nucleotides have crossed. So I will say the fragment size is 2. Next is, if deoxynucleotide is what is being added, now the chain elongation can proceed, is it not? Now, if the chain elongation proceeds, what I will get? Opposite to A, I will get T, again C, then G, then A. So at this point, you need to add A. So again, the termination can happen at this point. So what is another fragment that you will obtain? Another fragment you will get is fragment size 5. Sorry, 6. Fragment size of 6, right? So this is what is the so I will get fragment size of 2, fragment size of 6. Understood? So here, when in the first test tube, where you have added dideoxy ATP. Now, in the second test tube where you have added dideoxy CTP, if you are going to run the PCR, what you will obtain? You will obtain the first nucleotide that you add to this template strand is going to be G, and then followed by A, then T, then C. So at this place, there is going to be a chain termination or elongation. Two options. Chain termination happens whenever dideoxy CTP is being added at this place. Chain elongation will happen whenever deoxy CTP only has been added, right? So when the chain termination would have taken place, the fragment size would be 4. So I will get a fragment size. I will be having multiple fragments of fragment size 4. Now, if it continues, then what will happen? Again, G, A, T, C. So I will get a fragment size of 8 here, because at this point, at the last point only, it will undergo termination. Then next, if the third test tube undergoes this PCR. Now what is the fragment you will get? Yes, again, with this as a template strand, with this template strand, just write it down. Yes, what you will get whenever you are whenever this third test tube, that is dideoxy GTP, is undergoing. GTP test tube, wherever you have added that test tube, you are putting into PCR. Then again G. Yes, in the first place itself, it gets terminated. Yes, so I will get a fragment size of 1. Or if it continues, then what will happen again at this place? It gets terminated because you're going to add G. So I will get a fragment size of 5 here. Yes, similarly, what happens with the TTP test tube? TTP test tube again, I will write it here. So it will be a G, A, and T. So at third place, you will get it may get terminated, and if it follows, then at this place it gets terminated. Is it not? So I will get fragments of size 3 and also 7. Yes. So now what have I got? The first test tube, I've got few fragments. Every test tube, I've got few fragments of few molecular weight. Now I will be running a PCR. Yes, now I will be running a electrophoresis. Yes, now I will be running a electrophoresis. So along these 4 wells, what you will add? You will add this test tube, the dideoxy ATP test tube, dideoxy CTP test tube, then dideoxy GTP test tube, and then dideoxy TTP test tube. Right? So I will be adding this. Now what happens? So those fragments which are smaller will be at the bottom. The fragments which are larger will be on top. That's how the run is going to take place in this electrophoresis, right? So my fragment size of 1 is going to be at the bottom. Is it not? Fragment small, that is the smallest fragment size. So that will be at the bottom. So where do you have this fragment size 1? So along this the dideoxy GTP, followed by a little bit of higher number, which is the fragment size 2. So I will have this fragment size 2 a little bit above here at this first test tube. Then what happens? Third fragment is somewhere here. Fourth fragment is somewhere here, right? Fifth fragment is somewhere here, and sixth fragment is somewhere here, and seventh fragment is somewhere here. Understood? So based on the fragment size, it is getting separated in the electrophoresis. Understood? So now how will you now the fragment size? This is how the run has taken place based on the fragment sizes. Now from this pattern, electrophoretic pattern, how will you name it? How will you sequence it? So how will I sequence it? So I know this is the first one, right? I will know this is the first one. So G is the first one, followed by A, followed by T. Yes, followed by C. Yes, followed by G, then followed by A, right? And then followed by T. Yes, so GATC, GATC. This is how this sequence is there. So complementary, complementary to this strand that you have got is what is the template strand. So that's how you will interpret and conclude. Okay, so now we have sequenced our template strand now.